On behalf of myself and Dr. Weiner and the team here at Yale Cancer Center, it's really my honor to introduce our 15th Calabresi Memorial Lecture. And I think I've been here for about 12 of them in honor of Dr. Paul Calabresi. We always are proud to welcome the Calabresi family with us in person or online. We have actually in the front row Judge Guido Calabresi, and I'll have him say a few words at some point. And just received a week ago the honor of another Yale degree. He's already had several, and Paul's younger brother. And I've been very fortunate to know Guido for a long time as well. We also have Stephen Calabresi on his way from Rhode Island, and we have several other Calabresi family online. And we're really honored to have Stephen Rosenberg, this year's Calabresi lecturer. And Vince and I have been trying to get Stephen for years. And the offer of doing it virtually was fine because this is such timely work. It's the type of work that we want to build here at Yale. We actually have Mario Snoll here, will show up at some point, Steve. So we're really happy you're here. But let me say a few words about Paul. Paul Calabresi is often referred to as the father of oncology, and his influence here at Yale remains to this day. He's a former faculty member of Yale School of Medicine. In fact, I believe he held one of the jobs I hold. He built medical oncology at a time when I don't think there really was medical oncology. So he really built the field. He was internationally recognized for the pharmacology of anti-cancer agents. And if you walk through the B wing, you still feel his influence, the people he worked with, the people he recruited. And he also serves as the director of Yale Cancer Center's advisory board until 2003. Unfortunately, he himself passed away from cancer, showing us how, while we're making progress, we still have so much more we need to do. I had the good fortune to meet Paul, I hate to say it, 43 years ago. I don't think I'll talk about that too much here, but his son Peter was my freshman roommate at Yale. And I met Peter, and what better for someone who wanted to be a doctor to have as a roommate, the father of oncology. It was very nice. They didn't like me at first too much, but the family has warmed up to me over the years. And actually, I was looking through some photos last night. It was an era without iPhones, so it's hard to find photos like it is now, but I spent many memorable times with the Calabresi family. Over the Yale, Paul was an advisor, a mentor, and a really good friend to me. Actually, Vince and I still, we helped him with his disease as well. And having this job here means so much to me. One of the reasons I'm back at Yale, as Paul had always told me, Yale is a great place for you. I think you'd be happy there. So joining us today, we have Guido Calabresi, as I mentioned, Dr. Calabresi's brother. His wife, Anne, I believe, is online. I talked about Stephen, Paul's son, his wife, Mimi, and Paul's daughter, Janice, and son, Peter, are online. So this is a very special Grand Rounds. We have a plaque for you, Steve. I don't know if it arrived yet. It has to get through NCI security, but what I'm going to do now is I'm going to turn it over to Vince DeVita, who needs a little introduction here, but certainly another father of oncology and actually Paul's good friend. And I'm going to let Vince come up, introduce you, Stephen, and then we'll do a quick photo with you in the background. We'll figure out how to do a hybrid photo, and then we'll give the time to you for your talk. Vince.
And my welcome to the Calabresi family. It's always a pleasure to see you here and reminisce about my old friend and my mentor, Paul. I'm sure he's up there looking down saying, this is great. And it's a real pleasure to introduce my longtime friend and colleague, Steve Rosenberg. I met Steve in 1974 when he arrived fully formed as the new chief of the surgery branch right out of his training program. This caused quite a stir because the old administration of surgeons were old-fashioned, and their motto was, if you can't go wide, go deep. And Steve was a thinking surgeon, which immediately changed the ability to collaborate and to consult with the surgery department. It was a fun time He also came with his passion for immunotherapy. And in the book he was to write a lot later, he describes the source of his passion in immunotherapy. It was a patient he operated on in 1968 who came in with right upper quadrant pain, had a non-visualizing gallbladder, was all set to go to operation. And Steve reviewed his old charts, and he had come in 12 years earlier with belly pain. He was operated on, had big masses of tumor with metastases to the liver. And here he was 12 years later. He was supposed to die a few months later. He was 12 years later. He was fine. Steve thought this was a powerful expression of immunotherapy, and he wanted to find out how we could harness this work. And since then, with fierce intensity, he has worked in the immunotherapy field, and he has unwavering focus and stayed on this, doing work in vitro and then in animals, and then repeating the animal studies in humans to the point where he's become the lead investigator in the world in immunotherapy, having either discovered or developed all of the things that have been done in immunotherapy that are now making us very excited in the clinic. And since we're talking about fathers of programs, I would sort of view Steve as the father of the immunotherapy of cancer. He got his bachelor's degree and his MD degree from Johns Hopkins. He's got his PhD from biophysics from Harvard. He spent his time in Boston at the Peterbilt Brigham, getting a surgical training under the famous Franny Moore. And then during that time, he'd spent 2 years coming to the clinical associate at the National Cancer Institute. I also started as a clinical associate. We used to refer to ourselves as the Yellow Berets because there was a Vietnam War, and we got our military credit without any of the gunfire. Steve may forgive me for not going over his honors and awards. They're just too many. I stopped counting at 80. And finally, I've been working with Steve on the textbook, Cancer Principles and Practice of Oncology, for 42 years. He and I are 2 of the 3 editors, and it has been quite an experience. The reason the book has risen to the most popular cancer text in the world is the fierce competitiveness amongst the editors in preparing the book, making sure that the other guy was doing all the things he had to do. And of all the editors, Steve is the most competitive. And just a little story to illustrate that. The publishers took us away for each new edition 1 week before to set the table of contents and pick the authors and invite them. And at the end, the second week. And so we worked very hard during those days, getting up at 7 in the morning, working until 5. And at the end of the day, we would, do our exercise and relax. And Sam Hellman at that time played tennis. I jogged. And Steve, not so much. Steve wasn't really into exercise or a healthy diet at the time. So much to my surprise, at the end of one day when he said, "You mind if I jog with you?" And so I said no, with a little smile on my face. Off we went, me in my running shorts and my New Balance shoes, Steve in his khaki pants and tennis or sneakers more. And we started running, and after a little while, I ratcheted it up, and Steve ratcheted it up. And a little while, I did some more, and Steve ratcheted it up some more. And pretty soon, we were sprinting as fast as we could with our heads down, not to some specified goal, but clearly to see who would collapse first. And Steve Rosenberg, the unconditioned Steve Rosenberg, collapsed about 4 seconds before the conditioned Vince DeVita. And I tell you, ladies and gentlemen, that's the last time I beat Steve Rosenberg at anything. His talk today is entitled "The Lymphocyte, the Living Drug for the Treatment of Cancer." Dr. Rosenberg.
Well, this is a unique pleasure for me for several reasons. It's a pleasure to honor Paul Calabresi and his family. Paul, a giant in the field of medical oncology. but also to present in front of Vince DeVita, who has played such an important role in my ability to do a lot of this work. Vince is a true giant in the field of oncology, and his contributions are known to you all. But when I started working at the NCI trying to develop immunotherapies for treatment of cancer, there was not a lot of enthusiasm for it. But as I initially began to get results, I remember going to Vince DeVita and saying, look, I think there's something here, and I'm going to need more resources. And in a remarkably generous way, I was given by Vince DeVita, probably over the objection of many, I know over the objection of some others, was given the space and money and resources to conduct the kinds of studies that I'll be talking about today. And it made a huge difference to me. We became friends for the past 47 years. We worked in a textbook. Vince is the soul of that textbook, and has led the process for now through 12 generations of the book. And so to be here to honor Paul Calabresi, to honor Vince DeVita, Dr. Weiner, thank you so much for the invitation to come and deliver these remarks today. So I'll be talking about lymphocytes as a living drug for the treatment of cancer. The use of lymphocytes in much the same way that we might use chemotherapy or other targeted agents to administer to the patient to try to impact on the tumor. And I'll be talking about this particular kind of immunotherapy, and that is cell transfer immunotherapy. That has many advantages. One, if we're going to use a lymphocyte as a drug, we can grow lymphocytes easily to 10 to the 11 cells or more and administer very large numbers of highly selected cells. Because we have the reagent in the test tube, we can potentially identify the exact subpopulations and effector functions that are required for the cancer regression. And third, and very importantly, we can manipulate the host prior to the cell transfer in a way that you cannot do with other forms of immunotherapy, because the cells to be used are outside the body, and we can therefore alter the microenvironment of the tumor in ways that will enable immune cells to enter into those tumors and destroy them. And so it's this cell therapy that I'm going to emphasize this morning. Now, cells carry a conventional T cell receptor, and the entire function of the lymphocyte is dependent on that T cell receptor to recognize its antigen, which in conventional forms for CD8 or CD4 cells is a processed peptide coming from inside the cell and put on the patient's own MHC molecule. But a little over a decade ago, chimeric antigen receptors were described at the Weizmann Institute, and that converts a lymphocyte into the recognition of an antibody, not based on the conventional T cell receptor, but by making a single chain of the heavy and light chains of an antibody and connecting it to intracellular signaling domains. We can then use that lymphocyte to become recognition based on an antibody rather than on a T cell receptor. It can recognize cell surface molecules based on this antibody recognition. And so it provides us with a whole other way to identify targets. And I'd like to spend just a moment on CAR T cells, because although they've had substantial activity in the treatment of the hematologic cancers, they have not had activity against the solid tumors. And what's the reason for that? Well, CAR T cells require the use of monoclonal antibodies that recognize molecules on the cell surface. And they were described by Kohler and Milstein over 45 years ago. And despite extraordinary work, there's not been found a monoclonal antibody that can distinguish a malignant from a normal cell. The antibodies can have a lot of influence by reacting with cell surface molecules that can affect cell signaling, can affect cell growth. But We do not have antibodies that are unique to a cancer. And that's a problem because it's just as easy to kill a normal cell as a cancer cell. And we've seen deaths due to the application of cells that do not clearly distinguish between a tumor and a normal cell. Normal cells are highly exquisitely sensitive to the expression of monoclonal antibodies, and using ones that can attack normal cells have major clinical toxicities. And thus the limitation of CARs for solid tumors is substantial. And there are as yet no known CAR T-cell treatments that are capable of treating in a reproducible fashion malignant cells. They are, however, potentially very valuable for the treatment of hematologic malignancies. And back in 2009, we reported the first patient to be treated with a cell therapy that finally got approved by the Food and Drug Administration, the only cell therapy now approved, and I'll just spend a moment talking about it. We had developed it in animal models showing we could treat syngeneic tumors by targeting CD19, a molecule on virtually all B cells and B cell malignancies. We saw a patient with an aggressive lymphoma in the way it behaved. As you'll see as x-rays, he's had multiple chemotherapies, vaccines, checkpoint modulators, more chemotherapy, and finally came to us in May in 2009 for treatment with his own T cells that were genetically modified with a chimeric antigen receptor that could recognize CD19. And this is what his x-ray looked like when we treated him. You could see large masses directed by these yellow arrows in his mediastinum, in his axilla, large mediastinal mass, huge spleens, lymph nodes surrounding his vena cava and aorta, huge iliac vessels. We treated him. All of his tumor disappeared over the course of a few months, and he remains disease-free to the present time. He had bone marrow replaced, which also disappeared. But you pay a price because normal cells can also be killed, and B cells disappeared at a time when normal T cells and natural killer cells were returning over the course of the week and a half after the cell infusion. It took 8 or 9 months for the precursors that were not destroyed to restore B cells in the patient, but patients can survive for long periods of time in the absence of any B cells. Well, we treated the first 10 patients, 6 of whom responded, 5 of whom are still responding to the present day over 10 years later. And in the surgery branch, we received these kinds of results. We had objective responses by 47%, 42%. Are ongoing and have never recurred, out with median survivals now beyond 8 years. A good friend of mine, Ari Beldegren, had been in my lab 20 years earlier. We had remained friends, and he heard about some of these responses. And after we had 6 complete responders, he contacted me and said he wanted to start a company, Kite Pharma, who went on to do a multi-institutional study that almost exactly reproduced our results. We began interacting with Kite in 2012 through a research agreement, a research and development agreement, a CRADA, to transfer our technology to Kite Pharma. 5 years later, they received FDA approval, along with Novartis, who had begun working on this a year later. And in October 2017, Kite, who has started to do this from nothing, was sold to Gilead Sciences for $11.9 billion, and it's now widely available. This treatment's now widely available through the United States and Europe, and now beginning in Asia as well. I think a very proud example of how findings in an academic and a government institution can then get translated to help people in need. This remains, however, the only T-cell treatment that has been approved by the FDA, although there are several others that have shown effectiveness against multiple myeloma that have actually just very recently been approved. So here's the problem of oncology in the United States. There are about 600,000 cases. The solid cancers, epithelial cancers, comprise about 90% of all cancers that cause death in this country. About 10% are the hematologic cancers. And the devastating impact of this, as you can see, is that 1 in every 2 or 3 Americans of us will develop an invasive cancer during our life. And unless we can find better ways to treat it, about 1 in 5 will die of the cancer. And so the major challenge confronting cancer immunotherapy today is the development of effective immunotherapies for patients with metastatic epithelial solid cancers that cannot be cured by any available treatment and result in 90% of all cancer deaths. The checkpoint modulators have had major impact on some solid tumors like melanoma, renal cell cancer, patients that have mismatched repair genes, but the overwhelming majority of patients with the solid epithelial cancers do not respond, with only single-digit levels of response to the combined use of checkpoint inhibitors. So how can we attack these solid epithelial cancers? And I'll talk mainly about them. But here's a general cartoon of how we do this. We excise a tumor. If you follow me along clockwise, we excise a tumor. We grow cells to try to identify cells with anti-tumor activity if we can. We grow them selectively to large numbers. We generally infuse 5 times 10 to the 10 to 10 to the 11 cells and reinfuse them following a non-myeloablative lymphodepleting regimen with cyclophosphamide and fludarabine that will eliminate T cells for about 8 days before they normally recover. I'm going to talk primarily about the epithelial cancers, but we learned a lot from melanomas. And let me spend a moment with this lesson. We treated 192 patients with metastatic melanoma with some of these results that I first showed Vince DeVita when we had our first lymphocyte transfer that mediated regression of a melanoma patient in 1988. As you can see, we did treat 192 patients with their own cells, their own tumor infiltrating lymphocytes that we would grow out of the tumor. Those cells are a sink for tumor reactive cells. You can see our objective response rate by classic RECIST criteria, which is the criteria I'll use throughout this talk, was 56%, with a quarter of patients having complete regressions. Only 2 patients that ever had a complete regression have gone on to recur. The rest have remained disease-free, and of these 48 complete responders, only 2 patients required more than a single treatment. The cells are alive. They can divide up to 10,000-fold in the first 2 weeks after they've been administered. And as they patrol the body, they find deposits wherever the circulation exists. Well, here are our results in those. The overall survival rates, progression-free survival rates, were about 37%, but notice the complete responders very rarely ever recur. Somehow adoptive cell therapy appears to eliminate the last melanoma cell. And so the rest of the presentation will be on trying to find factors that we can use to treat not only melanoma, but the solid epithelial cancers. And the first question that we'll discuss are, what are the characteristics of the cells that mediated cancer regression in vivo, especially to these patients with melanoma that have undergone durable complete regressions? And to do that, we used a high-dimensional single-cell transcriptome analysis of up to 10,000 cells per patient, a very elegant single-cell analysis available 10x from Illumina. Well, because we had a group of patients that could respond and not respond, we utilized this single cell approach to identify the transcriptome analysis of patients comparing responders from non-responders, because for the first time, we had a group of immunotherapy patients that were showing this distinction. And when we break all of the lymphocytes and their transcriptome analysis using these UMAP or t-SNE analyses using a near neighbor analysis, you can identify 22 different kinds of lymphocytes based on their transcriptome, the messages that they express into proteins. And there turned out to be one cluster that seemed to differentiate responders from non-responders. And when we looked at the transcriptomic analysis, it turned out that only that cluster, cluster number 1, could distinguish responding from non-responding patients. If we looked at the expressed genes in each of the other 21 clusters, the responders and the non-responders were virtually identical, except in this cluster that was largely non-responders. It was only cluster number 1's transcriptome that could distinguish responders from non-responders. And it turned out that cluster 1 was highly enriched in stem-like lymphocytes that do not express CD39 and CD69, 2 molecules of lymphocyte activation and differentiation. Well, it appeared, therefore, that maybe these CD39, 69 stem-like lymphocytes were the ones that were most responsible for the melanoma regressions, because when we looked at the survival of patients receiving either very high or low total numbers of cells, there was no statistical difference in the outcome of those patients. But if we now looked at patients that got either high or low double negative CD69, 39 double negative stem-like cells, there was a highly significant difference between the cells that were the patients that were responding to not responding based on the number of these double negative cells that they received. Highly statistically significant. Well, when we looked at the properties of these cells, they were true stem cells. If you divide a lymphocyte population of TIL and facts based on CD39 and 69 expression, the double positive cells, when isolated and grow, will only reconstitute themselves, double positive. But when you take the double negative cells, they reconstitute themselves, whereas the double positive cells do not. They are true stem-like cells. When you take cells in one of our trials targeting NY-ESO-1 antigen, you can see in red, the double negative cells from the infusion continued to sustain themselves as they grew in vitro and were re-stimulated one time after another, whereas the double positive cells disappeared as they grew. They were not stem-like. They could not reproduce themselves with their own native reactivities. And in fact, if you look at the actual transcriptomes, it is the stem-like markers like KLF2, TCF7, CD62L that were expressed in the response-associated culture. And if you then took this back to the mouse models that we had initially studied, PMEL mouse model of melanoma, you can see that, in fact, these cells, when implanted and allowed to grow for 10 days before treatment started, peripheral blood, they grew. If you gave double positive cells, they had some weak reactivity. But if you gave double negative cells at 2 different concentrations, including this very low concentration of 500,000 cells, the double negative cells could mediate dramatic anti-tumor effects compared to the bulk populations. And so we could thus identify these stem-like cells that had a profound reactivity and published that about 2 years ago. And have been utilizing it. We'll talk about some of the results in the epithelial cancers, but very recently, and in this unpublished data, we found that we could actually make the double positive cells that were fairly weak work much better if we could give them a vaccine that was targeting the same antigens that the cells were targeting. And you can see here, if we take the double positive cells, which are the most exhausted of the cells, and give them, they do have some reactivity compared to the control. But when you give the double positive, these double positive cells in conjunction with a vaccine, you can now make them very active and take even 1 centimeter tumors, 20% of the total body, 5% of the total body weight of the mouse to disappear completely. And that's something that we're now, clinical trial that we're now initiating. So we know the kind of cell we want to use, but what did the TIL actually recognize that enables the in vivo control of the last melanoma cell? And the fact that we had seen specific regression of cancer in the absence of any on-target but off-tumor toxicities led us to believe that we were targeting something completely unique to cancers. And those were the targets of cancer mutations that we suspected were the targets of the TIL. And so again, to identify the target, we have to identify this small peptide that comes from an intracellular molecule or a molecule that's been ingested by the cell that can then be presented to the T cell receptor. And so about 5 years ago, we developed this particular blueprint for the identification of cells that were recognized by TIL that could mediate tumor regressions. And what do? If you follow me counterclockwise, we excise a tumor, isolate the TIL, and extract DNA and RNA from that TIL and do whole exome sequencing so that we could identify every cancer mutation that was present in that cell. And we do RNA-seq to identify all the mRNA molecules as well. We then take those cancer mutations as 25-mer molecules and either as peptides or as mini genes put together in a tandem structure to form a tandem mini gene and put it into a patient's own antigen presenting cell. Now that antigen presenting cell contains all of the MHC molecules of the patient. And if any of these cancer mutations can then be presented on the antigen presenting cell and recognized by the T cell receptor of TIL, that forms a signal in the lymphocyte that enables us to identify it because of upregulation of activation markers, and we could then grow those cells selectively. So again, the key is to make a 25-mer peptide with the mutation in the middle so that any peptide that could be presented on the MHC surface is included. It could either be the last amino acid of the peptide that's presented or the first one, but it has to be in this 25-mer. And the advantage of this is there's no need to do any predicted peptide binding. Every candidate peptide and all MHC loci are included in the screen because both have to be recognized, the peptide on the MHC, and there's no tumor cell lines necessary. And as you know, it's very hard to grow tumor cell lines for most of the epithelial cancers. This can be done within about 2 to 3 weeks. It takes 10 days to do the to identify all of the cancer mutation sequences, another few days to do the bioinformatic analyses. And so one has all this information available within 2 weeks of the tumor resection. Well, we started in melanoma and evaluated 86 patients. Those tumors have more mutations than most, 556 as a median. We screened every mutation that was expressed in RNA-seq, 15,000 mutations in these 86 patients, to see if any could be recognized by the patient's own autologous T cell. So we looked at 218 immunogenic epitopes. 85% of patients could recognize their own tumor cells based on recognition of these mutations. Interestingly, only 1.4% of the mutations could be recognized because they had been cleaved and also presented on the MHC molecule of that particular patient. 92% were CD8 cells rather than CD4s. And our first surprise, every neoantigen that we recognized, all 218, were unique to the individual patient's cancer and recognized by that patient. None were shared between 2 melanoma patients. Well, we then did this for 130 consecutive gastrointestinal cancers, screened over 15,000 of the expressed mutations. 1.3% were recognized, interestingly, half by CD8 and CD4 cells. And for the first time, we found an antigen that was recognized in more than one patient. It was a KRAS mutation restricted by a fairly unusual CW802 class 1 molecule. The other 209 epitopes that were found were all unique to the individual patient. True in breast cancer that we just published last year, 43 consecutive patients, 100 immunogenic epitopes, 2.1% were recognized of the mutations recognized, mainly by CD4s, and all were absolutely unique. And here is an updated, as of last September, study of 205 consecutive patients. And note, we're talking about the GI cancers, breast cancer, lung cancer, GU cancers like ovarian and prostate, and across the board, as you can see, about 70 to 80% of the patients contain T cells that would recognize their own neoantigens, their own cancer mutations that were presented on their autologous cancer cells. And of this 363 neoantigens we found, we only found this one KRAS that was recognized by more than one patient found on this particular screen. Now, an advantage of targeting mutations is its applicability to target multiple cancer types, because we're targeting mutations, and most cancers have mutations, some more than others. But let me show you examples of what we've been able to see in individual patients. Most do not respond. I'll show you the overall results soon. But here are examples of individual cancers that can respond. Interestingly, the first patient that responded to T cells that were unique, that were identified as uniquely responsive to our own mutation, it was an ERB2 mutation, was a 45-year-old woman with a cholangiocarcinoma, bile duct cancer, had undergone a hepatectomy, multiple chemotherapy regimens, developed lung and liver metastases. We treated her with unselected TIL, much as we did in melanoma. That does not work for the epithelial cancers. Unselected TIL do work in melanoma, but you have to select the specific ones, which are much rarer in the epithelial cancers. We gave her those. She didn't respond. However, when we used our tandem mini-gene approach, she had 26 mutations. We could found an ERB2 IP mutation that she recognized. It contained almost 90%. The infusion bag of cells recognized this mutation, and she underwent a complete regression of all of her cancer. You can see her lung cancers gone. She had 3 liver metastatic deposits that disappeared, and she remains now disease-free almost 10 years later. This woman who had a metastatic breast cancer had been through 7 different treatments for her metastatic disease to multiple groups, chest wall, bone, multiple nodal groups. She came to us, received cells for treatment. She received 4 different, what appear to be random somatic mutations. There's no driver function involved in these. And there was redundancy in the T cell receptors that we used to treat her. But by treating these 4 now random somatic mutations, she underwent a complete regression of this lesion beginning to grow through the cell wall. You can see multiple liver metastases. She had many more which disappeared, and she's over 5 years later now completely disease-free. This patient with a metastatic cervical cancer, though, was very aggressive, fungating into her vagina, underwent resection, radiation therapy, and cisplatin chemotherapy, underwent a hysterectomy and excision of both ovaries. She developed liver, lymph node, intra-abdominal mets, including one that was obstructing her ureter, came to us for treatment with our own TIL. You can see these lymph nodes which disappeared, this chest wall lesion disappearing, this one as well. This node was obstructing her ureter. We put in a urinary catheter, a ureteral catheter. When our tumor went away, we could take it out. She remains disease-free now over 7 years later. This patient with colorectal cancer was the one in which we found the KRAS receptor. It had a colectomy, was invading her bladder. So it was very aggressive. We resected 2 lung metastases. She had 7 others, treated her, and almost all of her tumors disappeared. She had 7 lesions, 6 of which disappeared. This one did not disappear and continued to grow. And when we resected it, we learned that, in fact, by looking at copy number analysis of the chromosomes, she had lost 1 chromosome from chromosome 6, and that chromosome encodes MHC molecules, including her restricting element. Therefore, that tumor could escape. When we then went on to resect her, that 1 lesion, she has not recurred since and remains disease-free over 6 years later. We can see responses in pancreatic cancer, as you can see this very dramatic response, which I show you. It was a very recent patient who had what appeared to be almost a complete regression of multiple liver metastases. But unfortunately, within 3 months, this patient did recur. And when we biopsied one of the lesions, he had lost expression of his target molecule, which turned out to be P53, and a molecule that we'll hear about in a few moments longer. Well, we've now treated a little over 100 patients with epithelial cancers. Again, it's the ducts in these organs that provide the source of mutations that are turning over constantly. And as mistakes are made, mutations appear, and those are the ones we're targeting. If you use bulk TIL in patients with epithelial cancers who are chemorefractory, we do not see responses in 21 patients. But when we started to select these TIL and treated 81 patients, 17% of them have responded. I've showed you some of them. These are all patients that are chemorefractory. Many had checkpoint modulators, which do not work in these tumors, and had not responded. We have a long ways to go, but these 17 patients at least show us that this is possible as we continue to refine and learn how to treat these patients with the refractory epithelial cancers. Well, there are two hypotheses that come from this. First, it appears to be the recognition of random somatic mutations. It's a final common pathway that explains cancer regression from most, if not all, immunotherapies. We finally understand what a cancer antigen is. And as we now look at the variety of immunotherapies, it's now been shown for anti-CTLA-4. We're studying it for TIL, tumor-infiltrating lymphocytes as well. What is a cancer antigen? It's any intracellular protein that could potentially be a cancer antigen if it's mutated and processed intracellularly to a peptide that can bind to the autologous MHC molecule. About 1 in every 70 of these mutated neoepitopes are neoantigens. And there's good news and bad news. The bad news is that this will have to be a very highly personalized treatment. After all, we're taking a patient's own cells, we're targeting a mutation that's unique to his own tumor, and will therefore be complex to administer. The good news is that virtually all cancer patients are potentially eligible because they all have mutations, and some more than others. So the opportunity does exist to further deliver this treatment. And the complexity will be difficult. But then again, I heard that in the early days of our development of CAR T cells, several groups came through, large pharmaceutical companies saying, hey, if we had this disease, we'd come to you, but we don't see how to make money doing it. But I have every confidence that if we can figure out ways to make it work in large numbers of patients, the genius of American industry will figure out a way to deliver it. Well, there are two main approaches to using lymphocyte transfer, and we've talked about expanding naturally occurring anti-cancer cells. But because now it becomes so readily easy to identify T-cell receptors, we can actually identify T-cell receptors into autologous lymphocytes and expand normal cells and convert them into anti-tumor T-cells. We've talked about these non-mutated proteins that are not on normal tissues, CD19, the unique somatic mutations, but there are mutations in cancer driver oncogenes or tumor suppressors that can be shared among patients. It's remarkable now that so many different cancer genomes have been sequenced, how few of these actually exist that are shared. Far and away, the most common are KRAS and P53. KRAS expressed in 30% of all cancers, 70% of pancreatic cancer. It's P53 in half of all cancers. And so we've made efforts to identify TCRs from patients that contain these mutations to find T-cell receptors by doing highly directed screening, using very high concentrations of these molecules, or especially by in vitro sensitization, to identify T-cells, to identify that very tiny number that do exist in patients that can recognize KRAS and P53. And we published about a year and a half ago a library of T-cell receptors that are CD8 and CD4 that can recognize KRAS, the common KRAS hotspot mutations. Over 80% of all KRAS mutations occur at 3 different hotspots: G12D, G12V, and G6 and the 61th amino acid. Almost all of them, however, the great majority are at this K12 and 13 position. And you can see for a variety now of restriction elements, we can identify T cell receptors and publish the sequences of them that can recognize tumors, mutations based on the recognition of KRAS mutations. And a similar library now of mutations in KRAS can be recognized by CD4 cells using a variety of different class 2 restriction elements. And if you look at the 2 libraries that we've now developed, 33% of all patients with KRAS mutations can potentially be eligible for treatment, these T cell receptors, if we can learn to use them. Well, that led us to the issue of, well, what kinds of receptors do we really want? Because we can find dozens of redundant receptors recognizing the same exact molecules. And there are a variety of tests that one can use to test these receptors: lytic function, cytokine secretion, the avidity, the affinity, catch bond techniques. And so we've gone to try to understand what T cells do we need so we can select the right ones among the redundant number. And this brings us back to that patient with KRAS who was treated with 4 different receptors, all that recognized KRAS. You can look here at their avidity, that is, they all recognize about the same concentration of peptide, but one of these receptors disappeared immediately upon infusion, and this was a majority receptor given, where our 3 of these receptors persisted well out beyond a year. Here are measurements out to 290 days. There was something very different about this receptor compared to these. What was the difference? The avidity was the same. We looked at a variety of criteria, especially surface plasmon resistance, to measure the exact KD, the association constant of that receptor. What we did is identify the receptor, clone it, purified it, and put it into human cells that were then used to treat the human tumor in immunosuppressed mice. And notice this one receptor had the highest affinity and was the least active in treating mice. If you look now at this highest affinity receptor using a human receptor to treat a human tumor in a highly immunosuppressed mouse, it was these lower affinity receptors which were the most effective. And so it appears that it's not only the fitness state of the lymphocyte itself, but the quality of its receptor that play a role in anti-tumor effectiveness. Well, knowing the receptor that was developed by Eric Tran, who was a fellow in the laboratory, who about 3 years ago moved to Portland. With Eric, we used this receptor that had the low affinity, that seemed to have that sweet spot of a recognition to treat a patient with pancreatic cancer. It was published in the New England Journal about 6 months ago. And you can see the regression that was reported with follow-up out to 6 months of multiple lung metastases, which shrank in that patient to perform a substantial partial regression. We have additional follow-up now. That patient did recur at 1 year, but spent 1 year disease-free of his pancreatic cancer. And we recently, just 4 months ago, treated a patient with pancreatic cancer utilizing a different set of KRAS receptors restricted by A1101, which is a class 1 MHC molecule. And you can see here this liver metastases, which has almost disappeared by 3 months, this large one, smaller, and by 3 months almost gone. We're continuing to follow this patient. But this is an approach using T cell receptors into normal cells that can potentially be effective. Peter Kim, in the surgery branch, a fellow, has developed a library mainly using in vitro sensitization to target p53 molecules. This was published about 6 months ago in clinical cancer immunology research. And again, these receptors, now with some common class 1 molecules, O201, can potentially treat about 5.5% of all patients with KRAS mutations. And again, 50% of all cancer patients have KRAS mutations. Well, we could again identify the T cell receptors that were most common in recognizing p53 and recognizing tumors that contain p53. We isolated those T cell receptors that can uniquely recognize p53, recognizing tumors. And again, studied each one of these 6 receptors that we could find to see which were most effective. And one of them was more effective than the rest, although at high concentrations, many others began to work as well in terms of causing the regression of a human tumor in a mouse using human receptors. At 1E7 cells, you could see several other receptors were active, but when you went down to a fifth of that at 2E6, a tiny number of cells, this one receptor was most effective, and it was the receptor with an intermediate affinity. And so as we continue these experiments, we're beginning to learn which kind of receptors we need. Well, having identified that, a patient who came in with breast cancer who had been through multiple chemotherapies with highly advanced disease was treated with our own cells that were transduced with a high affinity, a high avidity, but not a high affinity T cell receptor. She had very aggressive disease, including a pericardium that was replaced by tumor. We know that because a week before we treated her, we had to perform a pericardial window to release fluid from inside the pericardium. And all of the biopsies here were positive. She had pleural effusions. She had tumor covering her breast and extending into the other breast. She was treated with our own cells that had been transduced to express an anti-p53 receptor, this 175H receptor that I just mentioned. Every one of these nodules is a separate tumor deposit at a large necrotic lesion in her breast. All of this, everything visible disappeared. And you can see here at 60 days, the way this breast looked. At 6 months, however, she did recur with a nodule that we biopsied that had an LOH, a loss of heterozygosity at her MHC locus, which enabled this and other lesions to escape. And so she did recur at 6 months. So we can use T cell receptors to treat patients. And I'll finish with this very latest finding we just published about 6 months ago in Science and are beginning to exploit. And that is a very rapid method to identify cancer-reactive T cell receptors directly from a resected tumor without having to do all of the testing to see what they recognize. So how do we do this? We use a single-cell transcriptome analysis of lymphocytes from freshly resected tumor. In this analysis, each cell is barcoded with an individual DNA sequence. And when that individual cell is sequenced, and you can sequence up to 10,000 cells at a given time, the transcriptome, all the messenger RNAs can be analyzed and the T cell resequence identified, and each identified in an individual cell. And so we did that. And if you then break those lymphocytes into all of the different clusters, what you can do is the following. Because we have the transcriptome sequence for every individual cell, and we've identified the T cell receptors in that patient that can recognize the tumor, because every time we identify a cell in all the patients I've showed you, we can very easily then get to the T cell receptor using PCR techniques to clone it out. Only takes about 2 weeks. If we look at this cluster and look at the T cell receptor sequences that we've identified for this rectal cancer patient, and see what cells they're in, they quite astonishingly all appear in a single transcriptome culture. It was true for this breast cancer patient in this cluster. If we take 9 cancer patients from many different histologies, you can see they all fit in these clusters. And so what that enables us to do is identify the gene signature of cells in that cluster, because we know the whole transcriptome, all the mRNAs expressed, and could identify and report on a gene signature, which we published in Science, led interestingly by a B cell antigen, CXCL13. And when we take now an unknown patient cluster, look for that transcriptome sequencing, that we look for those T cell receptors sequences that fit this gene signature, we could then identify for unknown samples, we could identify cells that contain that gene signature. And because the cells are barcoded, we can immediately get to the T cell receptor sequence and know that within a few weeks of the resection. And when we test each of the TCRs in that signature right now, and we're trying to define that signature of the CD8 cells, 62% of all of the T cell receptors that are present in that cluster are tumor reactive, and we can identify it within weeks. In CD4 cells, it's not quite as good. It's about 1/3 of the T cell receptors. Thus, anti-tumor T cell receptors can be quickly identified without extensive screening and used for cell therapy. We haven't published much of this yet, but in fact, are working hard now to try to improve our ability to use T cell receptors for treatment. Well, I might conclude with this final slide and leave you with these few general conclusions. Cell transfer therapy can mediate durable regression in patients with metastatic cancer refractory to all other treatments. That T cells recognize unique somatic mutations in common cancers, that identification and targeting these mutations unique to each cancer, or sometimes shared mutations such as KRAS and P53, have the potential to extend cell therapy to patients with the common epithelial cancers using either these naturally occurring or T cell receptor transduced cells. And finally, gene signatures can be generated to identify anti-tumor T cell receptors in fresh tumors, as well as identify the phenotype of lymphocytes that can improve functions in eliminating tumor in vivo. Well, I thank you for your very kind attention.
Thanks, Steve. That was inspiring, and certainly I know there were a good number of questions. I know Diane Krause has a few online, but as is our tradition at the Calabresi lecture, we will often turn to Judge Calabresi or Stephen to please ask the first question.
Dr. Calabresi's son, and I'm a law professor, not a doctor, but I wondered, would the technique you're talking about have any applicability to glioma brain cancer, which I know are especially hard to treat, and which may become much more common in the future because there does seem to be some evidence that cell phone use increases the risk of coming down with brain cancer. So I just wondered, is this applicable to glioma brain cancers?
Plus brain cancers, glioblastomas, the most aggressive form of the brain cancers, do express mutations. We have identified mutations in glioblastomas. In fact, we published a paper on that by Ved Lako, who is a fellow in the surgery branch, of a mutation in a glioblastoma. But we have not treated any glioblastoma patients with these uniquely reactive cells. We have treated glioblastomas with CAR T cells targeting a shared mutation and saw no responses, again, because of the weakness of CARs and the potential danger that their tumors might express normal antigens. And in a separate study that I won't go into, we actually saw substantial toxicities by targeting a shared antigen in glioblastomas. But using the new cancer antigens that result from cancer mutations, I think should be tried in glioblastoma. We have not begun those studies yet and are concentrating on the more common epithelial cancers. But it's a wonderful idea and something that I hope to get to in a serious way soon.
Diane Krause has a question. I wonder if we can unmute her so she can ask it herself. While we're doing that, I'll just ask a question from an anonymous attendee. I have a patient with one of the targetable RAS mutations with the appropriate HLA. What can I do for them? How do I send them to you?
We're actively seeking those patients. And if you email me, sar@nih.gov, I'll see that you get contacted immediately about that patient to evaluate the eligibility of that patient for our studies.
Yeah, I'm guessing that was Diane Krause so that Eric would see that he has to put more resource into this so that we'll keep them here. He's laughing. Okay, Diane, are you able to ask your question online or do you want me to read it for you?
I can ask it. I can
Okay,
ask it. My question was why the selected TIL work when the bulk TIL do not for some of these patients with solid tumors. Is it a matter of the large dose of the effective TIL or potentially inhibition by other TIL that aren't targeting the cancer?
We have evidence for both, and I think both are important. In the animal models, the number of cells you give is very highly related to its effectiveness. In the human, although we generally give as very large numbers of cells, even within the numbers of cells we give, which generally are between 10 to the 10 and 10 to the 11, we do see an influence of the number of cells and the likelihood of having complete regressions. And we just published that about a year and a half ago. But we have evidence in animal models that the normal cells that you give can inhibit. Now, if you're giving normal cells that contain T regulatory cells, that would be hurtful. But also these other cells that you give that are non-tumor reactive compete for the cytokines that are a result of the lymphodepletion. When you lymphodeplete, you increase circulating levels of IL-15, IL-7, which normally do not circulate. And those circulating cytokines then can impact on the cells we administer. And if we're administering normal cells, they compete with the good ones. So you've hit your question actually hit on exactly the right answer that you mentioned. You need the right cells and none of the wrong cells.
Great. Thank you so much.
I know we're a little over, but just two more questions because some of your old friends, Mario Snow is online and would like to ask a question. Mario, you should have come here in person, Mario.
Sorry, Steve, I'm just curious, for all the reactive TCRs that you found in the epithelial malignancies, are those internally differentiated cells? Are there any in the stem cell pool that you say work well? And is that different between epithelial malignancies and melanoma?
We can find them easily in melanoma, and they're very difficult to find in the epithelial cancers because the incidence of those cells are likely 100,000-fold less in our measurements in the epithelial cancers than in the melanomas, because we generally try to find them in circulating cells. But they do exist. They're just very hard to identify. And my suspicion, as we continue to study and find better ways to identify tiny numbers of them, we will find them in the patients that did respond. But it's a particular delight to hear Mario, who worked closely with us for several years, as are several others of your fellows. We now have Nick Clement on our staff, and it reminds me to say that in fact, virtually all of this work, except for the first 5 to 7 years, was actually done not by me with my own hands in the lab, but by fellows who come to the surgery branch to train. Fellows like Nick Lemon, like Mario, who come to the NCI to gain experience in doing clinical and laboratory research for 2 to 3 years. And I owe them a great debt, as I do to Mario, for all the contributions he made when he was here with us.
You know, Steve, that might be a good way to end. We're going to move into the other room with some of our fellows to talk with you and with the judge and others. But let me just say, in hearing you say that, the Paul Calabresi I knew would have loved this lecture because it was innovative and it was patient focused. You're bringing new therapies to clinic, and Guido's going to say a word, but the fact that you mentored, that was what Paul is all about. And I'm going to give the final word to Judge Guido Calabresi, and then we'll retire to the other room. Judge.
Of course, one of the people who developed chemotherapy first, he said there was no doubt that where one went was not with chemotherapy, but with immune. And he said that was where it would have to be, and that's why I'm particularly delighted to have heard this little.
Well, on that note, thank you all. Thank you, Steve. We'll retire and we'll be 5 minutes and we'll get you back. Thank you, everyone.